Hey, welcome everyone. It's my pleasure to introduce today's seminar speaker, Colin Averill, who's a candidate for our ecosystems ecologist position. Colin received his PhD in an EEB program, the one at the University of Texas, Austin. And while he was there, he was also funded by NSF, both through a research fellowship and dissertation improvement grant. He moved on to Boston University, where he's a NOAA Global and Climate Change postdoctoral fellow. He also has been pretty efficient at trying to figure out our AV systems, both in chemistry and here. Let's see. So Colin's scientific interests, he's an ecologist who studies microbial communities in soil, and in particular how they affect global cycles. In particular, he's been looking at ectomycorrhizal fungi and their role in the global carbon cycle. Lots of carbon models assume an efficiency of 50%. That is, of the resource that a microbe uses, 50% goes into biomass and 50% is respired to CO2. Not a bad estimate for E. coli growing in rich medium in the lab. But there's a considerably bigger range when you consider microbes in soil. I think we'll hear a little bit about that today. He's also, in terms of thinking about interactions amongst organisms, put together an intriguing suggestion that game theory might be able to impart some insights, and I think we'll hear about that later on today. So join me in welcoming Colin Averill.
Hi, thank you so much for having me. Is this microphone working? Yeah. Great. So I've enjoyed everyone I've met with today. I'm really looking forward to the rest of my time here in Michigan. It's super beautiful. So I'm a soil microbial ecologist, but I work at both micro and macro ecological scales. And my work focuses on how soil microbes in terrestrial ecosystems regulate some of the largest fluxes of carbon on Earth. And in particular, how we develop ecosystem theory and ecosystem models that explicitly incorporate the ecology of soil microbial life. And to demonstrate you the importance of terrestrial ecosystems in regulating global carbon processes, here I'm showing a video of atmospheric CO2 concentrations from May to August. And what's really striking about this is that as the northern latitudes warm, where most of the terrestrial land surface is, you see this amazing drawdown of CO2. And that is regulated by biological processes, predominantly on land, exchanging massive amounts of carbon between the land surface and the atmosphere. And so you'd probably identify as photosynthesis as being important in that process. And you wouldn't be wrong. Plants convert CO2 in the atmosphere into biomass to grow. But a major component of what goes on in those processes happens below ground and is driven by microbes. And so through the processes of senescence and mortality, that reduced carbon fixed by plants enters soil organic matter pools. And at that point, that reduced carbon becomes food for microbial decomposers. And so free living microbes in the soil, so these are bacteria and fungi, exude enzymes into their environment to catalyze decomposition at the molecular level. And through that process, they gain access to carbon and nitrogen resources that they use for energy and nutrition. And also through that process, they regulate one of the largest fluxes of carbon on the Earth. And it's important to note here that this carbon flux driven by decomposition is almost equivalent in magnitude to all of terrestrial net primary production. It's also important to note here that this soil organic matter pool stores more carbon in it than all of terrestrial vegetation and the atmosphere combined. And so understanding, and that's just actually in the first meter of soils. Is that a meter? So understanding the processes that stabilize that carbon are really critical for forecasting the future of the terrestrial carbon cycle and of the Earth climate. Microbes also play the second important role with nitrogen in that plants require nitrogen from soils. It's locked in organic forms, and microbes decompose organic nitrogen and mineralize it into inorganic forms. And this is important because most of primary production on land is limited by soil nitrogen availability. And so this is just another way in which soil microbes regulate these important carbon exchange processes. So this schematic is basically the conceptual framework that terrestrial ecosystem ecologists use. It's basically the structure for most of our terrestrial ecosystem models. But plant ecologists in the past several decades have really enriched the ecological aspects of this framework. So by condensing plant diversity into distinct plant functional types, coniferous versus deciduous, C3 versus C4, they've greatly improved our understanding of ecosystem functions. We've recently been trying to embed trait-based frameworks and trait trade-offs nested within those plant functional types, further increasing the level of ecological detail incorporated into our ecosystem theory. And that detail is actually important because we can demonstrate that it improves quantitatively our predictions of the terrestrial carbon cycle. I think what's happening here is my computer is trying to connect to your network, and that's going to keep happening. And so I'm going to pause just to turn off my Wi-Fi, and I'm so sorry this is happening. And hopefully everything works again. Okay, so plant functional types, traits. There's an amazing opportunity to take an analogous type of approach with soil microbial communities. These organisms span multiple kingdoms of life, and new molecular technologies allow us to probe the composition of those communities with an amazing amount of detail. New metagenomic and metatranscriptomic approaches allow us to actually survey the distributions of traits in these communities by annotating those genomes and transcripts. However, despite this potential, the incorporation of ecological aspects of microbial communities and interactions and trait trade-offs into ecosystem theory is still at its earliest stages. So in my talk today, I'm going to address these things. Oh, I'm sorry. And one major reason why I think those are still in an early stage is because we're talking about interactions that happen at a molecular level, and we often observe them in a handful of soil. But then we need to relate those processes to the entire ecosystem, spanning multiple kilometers or scales of continents. Similarly, these interactions often play out over days to weeks, but we think they're affecting processes operating at decades to centuries. One of the most challenging problems in understanding ecosystem scale processes is scale. And so in my talk today, I'm going to address these different components I've discussed here. The first part of my talk is going to discuss my research on the ecology of mycorrhizal fungi in the context of the ecosystem, and specifically how I've been able to link both the presence and abundance of different microbial community members to changes in ecosystem function, both at very small and at very large scales. Second, I'm going to talk about my future directions, some of which involve incorporating aspects of evolutionary game theory into understanding the distribution of plant microbial interactions and ecosystem ecology, and then further pushing the scaling of soil microbial processes. And I'll do that by incorporating new genomic information, as well as applying tools not usually used by microbial ecologists. And those include taking advantage of forest inventory data, as well as remote sensing data. And so to begin, I'll be discussing these mycorrhizal interactions. And to do that, I'd like to very briefly re-summarize this ecosystem schematic. So we have trees here bringing carbon into an ecosystem. They turn over, they die. That reduced carbon gets stored in soils until it is consumed by microbes via the production of their enzymes and respired as CO2. But also, there's this important point that in most ecosystems, we think this process of nitrogen mineralization, microbes converting nitrogen from organic to inorganic forms, is really important for nitrogen uptake by plants. And that reflects the assumption that we believe microbes in soils are limited by carbon and not by nitrogen. And so if they take up something like an amino acid, but they already have nitrogen in excess, they may retain the reduced carbon in that amino acid, but they may separate it from the nitrogen and exude that nitrogen as a waste product in inorganic form, like ammonium. And this is the predominant way it's thought that plants access soil nitrogen resources, through this microbial recycling. And that reflects another deeper assumption, and that is that the microbial turnover of soil carbon is not limited by microbial access to soil nitrogen, again, because they have nitrogen in excess. I'm going to directly challenge this assumption. I think there's lots of places on Earth and in terrestrial ecosystems where this is not true. And I think where that's occurring really depends on which microbial community members are there, and specifically which functional classes of mycorrhizals are there. And to go there, I first need to introduce you to the mycorrhizal fungi. So these are fungal symbionts that associate with the roots of most plants on Earth. It's thought when plants first moved from water to land, really critical in that movement was association with mycorrhizal fungi, which basically acted as the entire plant root system. They're really important for taking up water and nutrients. And among the mycorrhizal fungi, there are two predominant classes of mycorrhizal fungi. And most plants associate either with one class or the other. The arbuscular mycorrhizal fungi are a very old monophyletic clade. Again, very important in taking up water. We think they're important with phosphorus acquisition. But what's important for my talk today is that as far as we can tell, based on genomic assays and functional assays, they either lost or never evolved the capacity to produce extracellular enzymes, specifically extracellular enzymes that target and catalyze the decomposition of organic nitrogen compounds in soil. In contrast, the ectomycorrhizal fungi are evolutionarily much newer. We think they've evolved over 50 different times, and we believe they've evolved from free-living decomposer ancestors in the soil. And as a result, based on genomic assays and functional assays, it seems that they've retained a lot of the capacity to produce these extracellular enzymes that can engage in these upstream nitrogen reactions. So they can actually break apart soil organic nitrogen like proteins, chitins, whatever form it may be in, and actually convert it into forms they can use. This distinction in enzymatic capacity forms the basis of what I'm going to be talking on from now on. And so just the really important functional distinction is the capacity to access upstream nitrogen decomposition reactions through the production of these extracellular enzymes. And so I would argue to you that this classic model of how carbon-nitrogen cycling works below ground is typical and probably what's going on in most arbuscular mycorrhizal ecosystems. Those trees rely on free-living microbes recycling nitrogen from here into inorganic forms to access it. Ectomycorrhizal fungi can play a very different game. So plants can allocate carbon, photosynthate, to these ectofungi. And these organisms will produce these nitrogen-degrading enzymes and take up nitrogen in organic forms and pass nitrogen to the plant. But what's really important here is that we had previously assumed this group over here, free-living decomposers, were not limited by nitrogen. And now they're in competition for the upstream nitrogenous resource. If that competition becomes strong enough, you'd imagine that actually nitrogen mineralization in organic end production would decline. And that's because these free-living microbes need to start retaining more of the nitrogen that they take up in their biomass to build more cells and grow. And if that nitrogen limitation becomes further strong enough, it may shift those organisms from carbon to nutrient limitation. And if that happens, that should downregulate their growth, their production of enzymes, and losses of CO2 that's derived from soil organic matter. And given sufficient time, that would actually stabilize carbon in these soils. And what's really fascinating about this is that this is a carbon storage mechanism that is not driven by climate, it's not driven by plant recalcitrant chemistry or soil mineralogy. This is driven by an ecological competitive interaction, operating between primary producers and decomposers for soil nitrogen. And it's mediated potentially by these ectomycorrhizal fungi. This is what makes this possible. In this first part of this talk, I'm going to present evidence to you to try and convince you and prove to you that this is actually happening. It's happening at the molecular level for the reasons I've stated here. And then we can observe signals of this competition and really large-scale patterns, and it has a major effect of where carbon is on Earth. Finally, I'll discuss some of my current work focusing on the biogeography of where arbuscular and ectomycorrhizal symbionts are, because if they are important for stabilizing carbon, then it becomes important to understand where they are now and in the future. And so to begin, I'm going to focus on field and laboratory experiments I've done testing the mechanisms of this hypothesis. And the first thing I'm going to tell you about is a field experiment I did trying to answer this very simple question. does the presence of this microbial functional group, ectomycorrhizal fungi, slow soil carbon and nitrogen cycling? And here's a picture of one of my field sites from the perspective of the litter layer. This is eastern hemlock forest. It's about 200 years old at the Harvard Forest in Petersham, Massachusetts. I work with eastern hemlock here. It's very nice because it forms these monodominant stands, so you have one tree species. It only associates with ectomycorrhizal fungi, and it partners very strongly. Basically, every root tip is colonized by these ectomycorrhizal symbionts. And in these forests, I did a straightforward exclusion experiment to test the effects of ectofungi. So I created treatments where ectofungi were either present or where they were absent. I did that by creating mesh bags, ingrowth bags, filled with sieved soil material. And so these are basically just small mesh bags that I fill with soil, and they vary in the size of the mesh. So the treatments where ectos are present are made of a 2 millimeter mesh, and that allows roots and ectomycorrhizal fungi to penetrate. And then the treatments that exclude ectomycorrhizal fungi are made of a 1 micrometer mesh. And that can exclude some of even the finest fungal hyphae that they are known to produce. And so here's a picture of one of them. It's one of the disturbance controls here. It's about the size of a window screen. And this is before I actually bury it. So I take these bags and then I insert them into the soil profile. And then I incubate them in the field for about an entire growing season from May to August. And the point of doing that is that gives sufficient time for roots and ectomycorrhizal fungi to grow into that disturbance control because they are not present when I first install those bags. And then, again, they'll be excluded the entire time in that 1 micrometer mesh bag. And then at the end of this incubation, I harvest these bags, I bring them back to the lab, and I incubate those soils to measure CO2 respiration, nitrogen mineralization rates, as well as several different enzymatic activities, enzymatic potentials in these soils. And my prediction is that carbon and nitrogen cycling rates will increase in those ecto-exclusion bags. And this will be because those free-living decomposers have been released from competition for nitrogen with those ectomycorrhizal fungi. And so in the context of the schematic I've been presenting, in those exclusion bags, I'm shutting down everything on the right side of this figure. And my prediction is this will amplify everything on the left side of this figure, all of these process rates. And so I'm going to move on to results now. So in green, I have the controls. In black, I have the exclusions. Here I'm plotting net nitrogen mineralization. And what we see is there's a huge increase in N mineralization rates. So this is the actual production rate of nitrogen mineralization, not the pool. And it increases in those exclusions, consistent with the idea that those microbes are now less nitrogen limited, so exude more inorganic forms of nitrogen. Second, here I'm plotting biomass-specific enzyme activity. So this is just total enzymatic activity, normalized per unit biomass of microbial biomass. And what we see here is that a more active microbial community that's decomposing more organic matter per unit microbe. This is soil respiration. This is just CO2 loss per gram soil carbon. This increases when we exclude roots and ectomycorrhizal fungi. So these soils are losing carbon faster when I remove this community member. And finally, I'm also plotting biomass-specific respiration. So this is just respiration normalized per unit microbe. This also goes up, consistent with like a more active soil microbial community that processes more carbon. Now, although all these process rates went up, a really key one went down. This is biomass-specific nitrogen depolymerization. This is the gross rate at which complex organic nitrogen in the soil and like proteins get converted into dissolved forms. And when I normalize it per unit biomass, it's an indicator, I interpret it as an indicator of microbial allocation to nitrogen acquisition. And we see it goes down. Again, consistent with the idea that they've been relieved from nitrogen limitation, so allocate less to these nutrient acquisition strategies. And so the presence of ectofungi in this system seems to be slowing soil carbon and nitrogen cycling. And there also seems to be evidence of this sort of enzymatic reorientation away from nitrogen-degrading enzymes and towards carbon acquisition, supporting this nitrogen competition mechanism. However, I also wanted to link this not just to the presence of these symbionts, but also their abundance. And to do that, I replicated this experiment in a different year. And I replicated it at a gradient of sites. So this is actually a stand age gradient that I believed would actually represent a gradient in the relative abundance of ectomycorrhizal fungi within the entire soil microbial community. And so the stand age gradient is all at the Harvard forest. My young stands are actually a girdled hemlock stand. It was girdled and killed to simulate hemlock woolly adelgid invasion. And for 6 years, it has been regrowing as ectomycorrhizal black birch. My intermediate age stands are the controls to that experiment. They're about 100-year-old eastern hemlock. And my old age stand is the one I've already described to you and where I conducted the last experiment. And I validated that this was indeed a gradient in the abundance of ectomycorrhizal and the ectomycorrhizal fungi within the total microbial community by first doing qPCR to quantify the relative abundance of fungi and bacteria within that entire microbial community. And then I sequenced the ITS2 region of the fungi, which is a fungal barcode region, to quantify the relative abundance of ectomycorrhizal fungi within the total fungal community. And I can use those two numbers and integrate them to get a metric of ectomycorrhizal fungal abundance within the total microbial community. And what I'm showing here is just that, yes, indeed, this does increase along this gradient. And so this age gradient is indeed a gradient in the abundance of ectomycorrhizal fungi within the soil microbial community. Again, my prediction here is the same as before, except that I predict that excluding ectofungi will increase microbial activity at sites with high but not low ectomycorrhizal relative abundance. And so here I'm plotting biomass-specific respiration again. Green is controls, black is exclusions. And we see again in that old growth stand, again, the same sort of increase in activity under exclusion. But we do not observe it in the low or intermediate stands. And so based on this, I conclude that soil carbon and nitrogen cycling is affected by both the presence and the abundance of this microbial community functional type. Now, since doing this research, I've tried to bring these systems into the lab, asking analogous sorts of questions, but with different ways of answering them. So what I'm showing here is a picture of these systems I've built. I call them ecosystem microcosms. I think they're really cool. Does this work? Yeah. So this is a tiny pine plant. This is a box. And this is a gas sampling lid. And this has a little rubber gasket here so I can plug these lids on and off. And what this allows me to do is to basically non-destructively sample respiration off these ecosystems. But what I really want to know in that respiration is actually how much, a lot of respiration that comes off the soils is from roots and recent photosynthate. And then the other half of it, and that percentage varies, is derived from free-living decomposers oxidizing soil organic carbon, breaking down soil organic matter. And so to partition that, I took advantage of a discrepancy in 13C isotope signatures. So pines are a C3 plant with a -26 per mil 13C signature. And I grow them in a soil I collected that has a C4 isotope signature. So this is a soil I collected from a native prairie remnant in central Texas that's dominated by C4 grasses. And so that's been in a C4 system for decades. And so as those plants die, the soil organic matter takes on the same isotope signature as the C4 photosynthesis, a -10 signature. And so basically all this allows me to do is to quantify how much of respiration coming off these ecosystems is derived from recent photosynthate and how much is derived from microbial oxidation of reduced carbon in these soils. And then, again, because they're in the lab, I'm just saying here that, yes, I can do this partitioning. I compare them with or without their ectomycorrhizal symbiont. So I can have roots in the tree present, and the only thing I'm changing now is the particular microbial community member. Before I had to exclude both the roots and the mycorrhizal fungi to do this in the field. And so I inoculate with Swillcoath ornatus. It's an ectomycorrhizal fungus. It also has a fully sequenced genome, which allows us to do more transcriptomic work. But basically, I create treatments where the ectofungus is present, where it's absent, or where it's present, and then I fertilize with inorganic nitrogen. And so I'm showing some preliminary data here. This is that soil organic matter-derived respiration off these chambers based on the 13C signature. And what we see is that in the controls without ectofungi, there's some rate of respiration. Now, when we inoculate with those ectofungi, we see about a 50% decline in soil carbon respiration that's derived from soil organic matter. And then when we add nitrogen, where those ectos are also present, we recover that rate of respiration, consistent with the idea that nitrogen is the mechanism by which these ectos are competing and inhibiting the activity of those microbial decomposers. To summarize this first part of the talk, both lab and field experiments support the hypothesis that ectos can slow soil carbon cycling, and this is likely mediated by a competitive interaction for soil nitrogen. Now, I do this in collaboration with molecular biologists at Duke, Ritas Vilgalis and Sunny Lau, and we're doing ongoing gene expression work in these, extracting RNA from these soils to try and get closer to the molecular mechanisms that actually underlie this competitive interaction. And so because we have the fully sequenced genome, we can actually take RNA and actually assign it to who made it, to some of those transcripts. So we could say, oh, this gene was upregulated by the ectofungus. This was probably upregulated or downregulated by the free-living decomposer community, to try and tie this to actually the mechanisms by which this occurs. And so to summarize, I presented you this framework of how terrestrial carbon and nitrogen cycling interacts below ground. I've challenged this that in ectosystems, there may be these interactions. And I believe I presented evidence to you that downregulates these processes, both in the field and the lab. But what I haven't shown you is that actually that leads to increased stabilization of carbon in the soil. And part of the problem here is that it is an issue of scale. It takes a long time to actually see and detect increases in bulk carbon storage in the soil. Even in plots where they do soil warming, where they heat up soils, which creates these huge effluxes of CO2 in the forest, it still can take them 5 to 8 years to detect changes in actual bulk soil carbon. And again, this is just a problem of scaling in ecosystem ecology. The actual drivers and the mechanisms are often far off from our ability to detect it in these bulk pools. And so in the second part of my talk, I'm going to try and show you how I've taken a different approach to try and identify the evidence that these ectos may actually be stabilizing bulk soil carbon. And to answer that question, I did another analysis, and I'm moving forward with the simple question: do ecosystems engaged in ectomycorrhizal symbiosis store more carbon in soils? And when I say more, I mean more relative to an arbuscular mycorrhizal ecosystem. And to answer this question, I did a literature and database search, gathering tons of soil carbon storage data to meter depth at 227 sites. And to include those sites, there needed to be, at the same place where that carbon information was taken, there needed to be a description of the soil, of the plant community where that was taken. And that's because with the description of the plant community, because most plants either associate with one type or the other, I can assign mycorrhizal association, whether it's ecto or arbuscular. And for inclusion, because I don't know much about a lot of these sites, but the relative abundances, we did not include any mixed ecto-AM stands. So these are all places where it's only predominantly ecto or predominantly arbuscular mycorrhizal associated. And my hypothesis is, again, always linked to this distinction in the enzymatic capacity, because ectos can engage in these upstream nitrogen reactions. And so then I statistically modeled soil carbon storage as a function of things I thought would represent inputs to the soil carbon storage pool and outputs to that soil carbon storage pool. So inputs, I have information on net primary production at all these sites. And for outputs, I included predictors that are often included in coupled carbon climate models that control decomposition in these models. And so that's strongly controlled in those ecosystem models by things like temperature and precipitation. Soil mineralogy, so finer particle soils like clayey soils are stickier, and that often leads to more carbon being stabilized, as well as vegetation type and biome type can be important there. And then I also included terms for the entire stock of nitrogen in those soil cores, because I also had that information, and then an interaction with the mycorrhizal association that's there. And my hypothesis is that after controlling for the nitrogen stock and after controlling for all these other variables that drive carbon storage, I would observe more carbon storage at the end of the day after statistically controlling for all these things in ectosystems. So what I found was that here plotting temperature, the relationship between carbon storage to meter depth on an aerial basis between temperature, precipitation, percent clay, and MPP. These relationships are very weak. I am not the first to report this. We know at regional gradients and at small gradients, these are important controls over soil carbon. But when you zoom out to global scales, these controls often break down. And again, this has been reported many times. And it's not to say that temperature doesn't matter. If you go heat up a soil in your lab right now, it will start respiring more CO2, I promise you. But again, at these global scales, this does not seem to be what's controlling these processes. Here I'm plotting soil carbon storage as a function of soil nitrogen storage. In yellow, I have ectomycorrhizal ecosystems, and white, I have arbuscular mycorrhizal systems. And what I see here is that these ectosystems are storing 40 to 70% more carbon in soils compared to the arbuscular mycorrhizal systems. And that effect size is after accounting for those other variables in the statistical model. And so what I conclude here is that these ectosystems, yes, are storing more carbon than the AM systems, and it's independent of those other drivers, and that the interaction is consistent with the idea that the ectocompetitive interaction may be driving this effect. And so now I'm going to move on to the final part of my talk about ectomycorrhizal fungi, and it's about understanding the geographic distribution of these organisms, their biogeography, because if they are important for stabilizing carbon, then it's important to know where they are now and where they will be in the future. And so in mycorrhizal ecology, there's this general hypothesis, and it's that inorganic nitrogen inputs to an ecosystem will favor arbuscular mycorrhizal-associated plants at the expense of ectomycorrhizal plants. So here I'm just plotting the relative abundance of ecto within an ecosystem as a function of nitrogen in, and it goes down. And the basis of this hypothesis is that ecto-associated plants spend a lot more photosynthate on their symbiotic partners. They engage in these upstream nitrogen reactions to access soil organic nitrogen, and these seem trivial if I'm giving you free inorganic nitrogen by fertilizing you. This is a surprisingly challenging hypothesis to test, because you can go out to a forest and you can add inorganic nitrogen, and you may see arbuscular and ectomycorrhizal fungi, the particular microbes in the soil, respond in the ways you might expect. However, it takes a long time for plant community composition to turn over in ecosystems. If we talk about forests, that could take decades, if not longer. And so actually knowing if this is changing the composition in a major way, because different tree species host different fungi, is a surprisingly challenging question to answer. I've taken a different approach to answering this question by going to the Forest Inventory and Analysis database. This is collected by the U.S. Forest Service. They go out and census forests all over the country for the purpose of monitoring forest productivity for economic purposes. But it's an amazing ecological tool. I used 3,000 sites in this database that newly have soil carbon data at 30 centimeters depth, as well as other soil chemistry data. And so this is a subset of the total FIA database. And I'll point out there are conspicuous states missing, like Wyoming and Oklahoma and other states. And that's because the FIA is administered at a statewide level, and this is new data, and some states are slower than others to report their data. That's the only reason those are gone. And so I'm using the data I have. And so there's these 3,000 plots. They remeasure the same exact plot every 5 to 15 years. And these are newer observations. And so when a forester goes out for the FIA, let's say this is a whole plot, and within that plot there are 4 trees. They'll go out and they'll measure the diameter breast height of all these trees, and they'll also identify them to species. I have a key that matches each tree in the United States to which mycorrhizal association it makes. And so I could turn this species information into mycorrhizal information. And so I can get microbial data from this forest inventory. And that allows me to calculate the relative abundance of ecto and arbuscular mycorrhizal associated trees based on their relative basal areas. And because they measure these plots through time, they go back, generally as trees grow, so I get growth rates of these different classes of mycorrhizas. Sometimes new trees cross a diameter threshold to be counted, so I get recruitment. And sometimes trees die, and so I get mortality rates. And then I can statistically model all of those processes as a function of climate and soil properties. And also, but I really want to know how nitrogen inputs affect this process. And I can do that because there's massive amounts of nitrogen deposition in the United States. So we burn fossil fuels, either in coal-burning power plants or in cars, and that releases NOx gas into the atmosphere, which then rains down as inorganic nitrogen on our ecosystems. And because there's heterogeneity in both sources, winds, precipitation, and topography, we get this semi-heterogeneous distribution of nitrogen deposition across the landscape. So I can take this nitrogen deposition information and layer it on top of the FIA database to ask this question. Very briefly, I'm going to touch on some of the models I used because they're slightly more complex than the mixed models and general ANOVA regression frameworks I've used so far. These are all Bayesian models, and they're Bayesian models only because it allows me to fit a slightly more complex likelihood function to these probability distributions. I like to keep it simple when I can, but this allows me to account for nonlinear dynamics in forest dynamics as they grow. And so we model the fraction of trees that are ecto with a beta probability distribution, recruitment as a zero inflated Poisson, and growth basal area increment at the stand level using just a log normal distribution. And all of these we model as a function of climate and soil properties and nitrogen deposition. And based on the nuances of the particular analysis, sometimes we need interactions with ectos based on the response variable. And I'd be happy to talk with anyone about this afterwards. But my prediction, again, is that inorganic N is going to reduce ectos in favor of arbuscular mycorrhizal fungi. I should see this. What I'm about to plot for you is the mean response of this relative abundance metric as a function of nitrogen deposition. And this is model output now. So this is after it's been fit to the full data set with all the parameters. And so this is the mean and the 95% credible interval around the relative basal area as a function of nitrogen deposition. And so things like climate and soil chemistry and other things affect the relative abundance of ectos and AM species. And what I'm doing is they have all their own parameters. And what I'm doing is I'm holding all those other predictors at their mean in the data set. And then I'm just turning the nitrogen deposition knob across its range in the data set from low to high. And we see this crash in the relative abundance of ectomycorrhizal trees per basal area. If we go and look at the underlying demographic processes, here I'm showing growth. AMs are in blue, ectos are in red. We see that nitrogen deposition increases the growth of trees. This is something we should expect. We expect MPP in terrestrial ecosystems to be limited by nitrogen availability, but it has a much stronger positive effect on AM species than ecto species, such that those curves cross. If we look at recruitment data, we see a negative effect on ecto species, but a positive effect on arbuscular mycorrhizal species. And so I conclude from this is that nitrogen deposition is shifting the composition of US forests, which we've known before. But this is suggesting that one aspect of the way it's shifting the composition of those forests is by selecting on their fungal microbiome, favoring these arbuscular mycorrhizal trees at the expense of ectomycorrhizal trees. And because we know that there's these strong effects of the abundance of these mycorrhizal groups on carbon storage, this likely represents an undocumented loss of carbon from the North American continent. And so to conclude this first part of my talk, I've shown you how microscale interactions can alter process rates, how these interactions between many different groups of complex microorganisms can be better understood through the lens of functional types, and that can lead to changes in process rates. And then I've used that information, that primary understanding, to make predictions and test predictions in macroscale processes and patterns of carbon storage data, of tree growth data. And the idea here is being able to walk across scales, translating the microscale molecular interactions to macroscale patterns and process. And so with that, I'd like to move on to my future directions part of my talk. And I'm going to start with talking about evolutionary game theory. And to do that, I'd like you to imagine how a tree should allocate above and below ground to above ground structures to get carbon and below ground structures to get nutrients. And I promise this will tie into mycorrhizas very soon. Now, I would argue to you that if I knew everything about the resource supply rates above and below ground, and if I knew everything about the return on investment in each of those resource acquisition strategies, and everything about the stoichiometry and physiology of this tree, I could write you an equation for how that tree should allocate above and below ground perfectly. And that would maximize its growth. And if growth is correlated with fitness, then that will also maximize its fitness. And that tree will do great. But that will only be the best strategy, assuming it's the only tree in the population. But we know other trees, and that's why I have two here, can interfere with other allocation strategies and compete for resources. And so to discuss this, I like to visualize this as a payoff matrix. So here, the payoff here, the first number is for the tree on the left, the second is for the tree on the right. They're both playing this optimized strategy that I just described, and we're assuming productivity and fitness are correlated, so each gets 5 units of productivity and 5 units of fitness. There may be a competitive overinvestment strategy, such that, let's say, this tree allocates more below ground to roots and takes up more nutrients, and it comes at the cost of their competitor. Now, it's not an overinvestment in such that it's a better payoff. So, either tree 1 or tree 2 could do it, which is why you have 2 different numbers here. But it's an advantage because through time, that first tree will then eventually replace that second tree in space. And so this will be selected for as it increases that tree's fitness, this strategy. However, eventually that will select for all trees in the population taking on this strategy. And that may lead you to a situation of the tragedy of the commons, such that everyone's over-allocating below ground, and it comes at a fitness cost to everyone. And everyone would be better off if they cooperated, they'd have higher fitness and higher growth. However, because of the landscape of the payoff matrix, it always leads to this tragedy of the commons, and this is selected for. This is called the evolutionary stable state, which was coined by John Maynard Smith and developed by George Price, and was really important for understanding ideas in animal conflict. It's since been very important in iterated versions of this game. This is also referred to as prisoner's dilemma or the hawk-dove game, have been critical for understanding the evolution of cooperation. And it's since been applied in macroeconomics to understand human effects on economic processes, but it's barely been applied in ecosystem ecology. But I think it really has a lot of potential to explain why things are where they are, how they allocate, and potentially also the balances of carbon in ecosystems on the Earth. And I think one way that connects to mycorrhizal ecology is that I think association with mycorrhizal fungi is an overinvestment strategy. I think it's a competitive overinvestment below ground. I can show you data that these mycorrhizal fungi will slow nitrogen mineralization rates. And all they're doing is just getting upstream nitrogen instead. They're paying for something they could be getting for free. But because they do that, they can competitively exclude an arbuscular mycorrhizal associated tree that relies on that free-living nitrogen mineralization to access those resources. So it may cost them, but so long as it competitively excludes their neighbor, it'll be selected for. And so why is in the world all ectos? So under low nitrogen situations, I think the payoff landscape is something like this, leading to a tragedy of the commons for ectos. But there's also situations where the ectos cannot sufficiently draw down that N mineralization rate, or there's other factors that affect that, such that under high nitrogen conditions, the payoff matrix may change such that AM are selected for. And because they use that free N mineralization, they don't pay this extra cost. They just always do better. And so that becomes the evolutionary stable state. I'm really interested in testing this hypothesis, and there's really not a lot of work investigating about this being possible. One way I'd like to do it is actually building these payoff matrices. And I would like to do that using the ecosystem microcosm boxes I've already described, but modifying them to have 2 trees that can interact and compete and do all pairwise comparisons of AM, ecto, 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 arbuscular, arbuscular. And then monitor ecosystem fluxes of carbon and nitrogen, and also manipulate nutrient input rates to those ecosystems. Second, I'd like to move to the field asking about neighbor effects. One prediction of this hypothesis is that an ectomycorrhizal associated tree should do better around a heterospecific mycorrhizal partner. So ectos should do best when they're around arbuscular mycorrhizal associated trees. And as they come to dominate a forest, they should start doing worse when they're around ectomycorrhizal associated trees. And it'll be really exciting to test some of these ideas at the University of Michigan Biological Station, where I can imagine a lot of these processes happening over successional time. You have explicitly manipulated succession there, and so that gives a lot of traction to answer those questions. And also, it's somewhat local, and we can get to freezers very quickly, and so we can actually probe the underlying molecular interactions that are driven by the microbial communities that cause this process to happen. And then finally, focusing on scale, I can imagine asking these questions and seek to answer these questions using large-scale forest inventory data because they have information about the relative abundance of these different mycorrhizal strategies within a community. Second, I'd like to just talk briefly about scaling microbial processes to the ecosystem and to the landscape. And to do this, I think it's useful to think about some relationship between microbial activity and some environmental driver. So generally, as soils get wetter, microbes are more active. When it's really dry, they're really stressed out, their enzymes can't diffuse to their substrates, they They're under osmotic stress, and as it gets wetter, they're happier and they work better. And it's really easy to collect data to show this. So here's data I collected across a rainfall gradient in central Texas. I'm plotting total enzymatic activity as just an indicator of total overall microbial activity as a function of soil moisture. And you can fit a curve to this, and it would be pretty tempting to go, okay, microbes move more when it's wetter, and so I'm going to make an ecosystem model, and I'm going to predict decomposition and microbial activity as a function of soil moisture. And this abiotic control is there, and it captures this process. And it wouldn't be a bad idea, and that's how it is done. But again, I collected these data across a rainfall gradient. So places where it's historically dry, but I chased rainfall events, so I caught them when they got to this soil moisture, and places where it's historically much wetter. So varying from 400 millimeters with the red dots to 800 millimeters with the blue dots. And if you put that interaction into this model, you see that they respond very differently based on their historical rainfall pattern. And so the ones from the dry places actually ramp up much faster. I liken it to if a desert receives a rare rainfall event, and all of a sudden you see trees burst into bloom and start growing very quickly. It seems like microbes may be doing something analogous here. I don't know if that analogy totally holds. And so, but the point is, if you parameterized what you thought the dry end of this gradient would do with that common relationship, you'd certainly get your number wrong under a no analog condition. But this was labor-intensive data to collect. I had to go chase rainfall events across this rainfall gradient over several hundred miles. I had to get this enzyme data that's not stuff that people collect everywhere. What would be great is if we had some proxy of traits that could place these things along those different trait axes. And there's an opportunity to do that with soil metagenomics because we can functionally annotate metagenomes. And you can imagine we could find genes associated with rapid growth and genes associated with osmotic tolerance. And those may give us the latitude to place those communities along those positions in trait space and explain the parameters of that distinction and how they ramp up and that interaction with soil moisture. And if we could do that, we could take advantage of things like the Earth Microbiome Project, which is collecting metagenomic data and aggregating it from places all over the Earth. And so I might be able to go to a new place where I haven't done this intensive sampling and say something about where it lies, that microbial community lies in trait space, and what that means for how it'll probably function under different conditions in a much more accurate way. Another way this data is being generated is with the National Ecological and Observation Network. These are permanent plots all over the United States. You've probably heard of them. And they're making metagenomic observations in soil every 3 months. And they're measuring lots of environmental variables that may allow us to look for and separate out some of these important trade-offs that we may be able to predict from soil metagenomes. Second, I'm really interested in working with more forest inventory data. I've used it in the United States, but there's forest inventory data sets all over the world. So there's a lot of traction in these data sets. And second, I've used them for mycorrhizals, and I'll certainly continue to do that. But any microbial functional type that is tied to the presence of particular plants could be analyzed through this lens. So if there was certain rhizobium or leaf endophytes that were specific to certain species of plants, an analogous type of approach could be taken with these forest inventory data. Second, I'm really tempted by some new papers coming out from NASA JPL suggesting that we can sense mycorrhizal types from space. So with forest geoplots, which are these massive forest inventory plots, you have one at the Edwin S. George Reserve. They map out in this huge spatially expansive way the relative abundance of AM and ecto trees like I've done in the forest inventory data set. And they have multiple blocks within a given site, and they have 3 sites they've done this with in the United States. And they can use the data from Landsat, the Landsat bands, there's like 9 or 13 of them or something. And they can also go on the ground and ground truth what's there. And they find they can predict the relative abundance by basal area of ecto and arbuscular associated trees with 85 to 90% accuracy, which is scary high. And I think we need to figure out what's going on there. There's probably some correlation between foliar traits and mycorrhizal associations. You might think it's coniferous versus deciduous, and coniferous is more likely to be ecto. But even in plots where all the trees are hardwood, this still works. And so this presents 2 exciting opportunities. One is that it spatially increases the coverage of where we know where these mycorrhizal types are in this enormous way. But second, Landsat's time machine, We have Landsat images that go back 30 years. And so it creates this opportunity to do microbial ecology in the past, taking advantage of unintentional disturbance experiments. So maybe there was a clear cut. Maybe someone fertilized with a bunch of, some pollution. But lots of different ways in which we can go back to the landscape in time and extract microbial information and see how it changes in response to some unintentional experiment. And my goal with all of this is this idea that maybe we can build these genomic trade-off signatures and embed them into bottom-up microbial ecology models that predict decomposition, and then constrain those models with macro-scale microbial data from these many disparate sources of information. And ideally, what I really would like to see this go forward, and I think we're moving towards, is that doing what plant ecologists have done for decades in incorporating plant functional types, but doing an analogous approach with microbes and decomposition, thinking about the functional types, thinking about trade-offs, and ideally creating a more synthetic ecosystem ecology that acknowledges all domains of life and their ecologies, and developing a more predictive ecosystem ecology. And with that, I'd like to say thank you so much. I need to acknowledge funding from NOAA, who funds me right now through my postdoc, the National Science Foundation, UT Austin, and Boston University. Thank you so much for your attendance and your attention. I really appreciate it, and I'd be happy to take any questions.
So I know a few people have to leave, but I mean, you did it. You gave us microbial life and ecosystems.
Thank you. You didn't believe I could do it? Sure.
The stuff that's making its way through the review process. And it seems to me that the information that informs your models comes from this cross-space analysis. You've got data on all these different ecosystems, different communities at different stages in succession and so on. And then you're extrapolating that to sort of look at what would happen with the change in nitrogen deposition in time. So there's a kind of a space for time substitution that you're performing here. I'm wondering at what point, what is it about that gives you, confidence that you can make that kind of substitution?
Yeah, so it's certainly a space for time substitution when I look at the relative abundance data and how the nitrogen deposition affects that relative abundance. That's a static measurement. What gives me confidence in that is that there's also temporally explicit data analysis. So this growth and recruitment data are not space for time. Their time is time because that's based on two measurements. And so we have the actual growth rate and the recruitment rate at all those places. And so those underlying demographic processes through time give me confidence that what I'm seeing in response to nitrogen deposition is actually changes in the demographic processes that would lead to a change in the composition of those two functional types.
When you're constructing your ecosystem microcosms,
Mm-hmm.
how do you get rid of mycorrhizal but leave bacteria?
Yeah, so those are fully live soils. So they have bacteria and fungi. They're free-living decomposer fungi in the soil as well. And it's actually really hard to go out and collect a soil and even autoclave it and then kill all the ectomycorrhizal spores. Why this works is because it's from this native prairie remnant, and that's dominated by C4 grasses, which are all arbuscular mycorrhizal. And so it's been a long time since that soil has seen any ectomycorrhizal symbionts.
So that was really cool stuff. I'm trying to get my head around the competitive dynamic.
Mm-hmm.
So, way back in the beginning of why the ectomycorrhizal fungi are superior competitors for nitrogen to the free-living decomposers. Because I think you are assuming that. It's not simply a symmetric competition. That those are better. And part of the answer may be, another part that I wasn't clear of, is you have the ectomycorrhizae going to the dissolved organic carbon, whereas you had the primary the free living are actually sort of
up that schematic real quick.
Those two things, I think need to understand that a little more.
Yep, that's totally fine. So they're both producing enzymes. I'm sorry, this font is different because this one inhibited them. They're both producing enzymes. What's distinct about them is that these guys previously were carbon limited, and their carbon resource is the soil organic matter. These ones get carbon from this plant. And so their primary reason for producing enzymes is to access nutrients. And so these ones can become nutrient limited if this one invests enough in resource acquisition. And assuming declining return on investment in any resource acquisition strategy, more overall strategy means less per capita return. And that means they are less efficient in acquiring nitrogen.
You mentioned that there's 50 independent origins of mycorrhizal fungi. Right now, but in this discussion, we're putting them in one box.
Mm-hmm.
Is it a resultant box? Do you have all 50 in there? Or do you have just 2 or 3 of those 50?
It depends on where I am, right? Like in those ecosystem microcosms, I have one. And that's one origin, right? And there's many hundreds or thousands of ectomycorrhizal species. And there's certainly variability in what ectomycorrhizal fungi do. I am certainly lumping a lot, and there's more function that could be split out here. We know that different ectos have different foraging strategies. It seems that the ones that actually forage more and create more diffuse hyphae and web-like hyphae play this strategy more. There's other weedy ectomycorrhizal fungi that don't seem to do a lot. They actually might be parasites on these plants. They're just sort of surface hyphae. And so yeah, there's certainly a lot of latitude in the functions of those organisms, much more so than I've represented in this talk. Either. Both of you have already asked questions.
So going to your sort of future directions and thinking about competition from a game theoretic point of view, I noticed you focus on below ground competition for nutrients amongst the plants, and it's very interesting game there. I wonder, though, of course, plants famously compete for light, and I wonder.
Heard of it.
How do you incorporate those aspects of the competition into that framework? Is that fundamentally changing or changing in a superficial way?
I think it's important, and I think it's important based on a lot of the work done by Steve Bacala, Ray Dubzinski, and Carolyn Ferrier, working on game theory in the context of above and below ground allocation, mostly in a root-shoot way. And they make this argument that actually most of the trade-off in above and below ground is not necessarily shading per se, but once you close a canopy, and all you have to do is keep up. And really, the weapon is the wood. So yeah, I think that would certainly be the case. I basically am relying on their work, which they make the argument that actually once you close the canopy, the real important aspect of competition shifts below ground. I'd be happy to dispute that, and it's very possible that they are wrong. But regardless, I think a component of that competition is going on, and not necessarily all of it. Certainly plants do more things than get nitrogen.
So that also, God, Aaron, we're on the same train here. I'm also thinking about the game theoretic part. The one interesting thing about the ectomycorrhizal fungi is that they're often thought to connect all the trees by being in, so you actually in some ways have a single organism where all the mycorrhizal trees are connected through the mycorrhizae. Does that need to play into thinking about this game in any way?
So you're talking about like the common mycorrhizal network research?
Yeah.
Yeah, that stuff is wild. They're talking about carbon transporting between different species of trees and nutrient transport between very, very different species, like pines and birches, co-located in the same place. Like arguing that like there may be selection for the superorganism of the forest.
Right.
That is tenuous. So yeah, they had that really nice science paper out this year, and it was great. And there was a Radiolab episode about it, which was also awesome. It's hard to believe it. Still, it's
simpler version of it. Just sit there